Hello, everyone. My name is Matt Elliott, and today I'm going to be presenting my lab's work in using Jupyter as a brain-computer interface. We colloquially call the platform that we created WetAI, as in artificial intelligence on wet lab biology. So the key highlight from what we are doing is that we are running closed-loop algorithms on real human brain tissue. So what that means is that we are reading neural data off of the tissue. Then, with algorithms in Jupyter, we analyze that data and perhaps update the algorithm. And then we then create neural responses. Essentially, we send messages back to the brain tissue. And you see here, we have a feedback loop, a beautiful feedback loop. So this algorithm is adapting to the brain tissue. But just as importantly, the brain tissue itself is adapting to the algorithm. My goal is to understand how the brain computes at a cellular level. And I believe this type of setup is fundamental to answering that question. So let me tell you a little bit about myself. I am a PhD at the University of California, Santa Cruz. My group is called the Braingeneers, where we combine engineering with neuroscience. It is led by David Hausler, who is recognized for assembling the human genome, being the first to assemble the human genome. You may remember the Human Genome Project and the race to assemble the genome between industry and academia. So he was the person who won that race. Let me tell you just a little bit about how I have used Jupyter in the past. I have been a major promoter of Jupyter software. I think it's been probably 5 years now since my master's. But after my master's, I was really excited about Jupyter, and I started a YouTube channel where I created tutorials teaching people how to use Jupyter. And I did pretty well at it. I got, over 5,000 subscribers, over 500,000 views. We can even watch just a little of it as an aside. It's just, this was a long time ago. So this was, I think, before Voila. So I created my own software for, just hiding the code so you could have an app. And this is a Jupyter notebook. These are just widgets. I think at the time I was creating a graphical model software. And, it's fun showing other people, teaching them how to create the same software in Jupyter. So you just get an idea that, yes, I'm a neuroscientist, but I was a coder in the past. Other Jupyter projects, at around that same time, I created something called Io, because Io is a moon that orbits Jupyter. And the purpose of that software was to make Jupyter more accessible, so to allow it to run on iPad and other tablets. And I put that on Docker Hub, and it became wildly popular, more popular than I could have ever imagined. It was downloaded over 5 million times. And then finally, also during COVID, I created a little website during the first 1 or 2 months of COVID, when it was still totally new. I created a website in Jupyter and posted it up. It also shot off like fire. I think that was 40,000 clicks a day when I created that, just because people wanted information about what COVID was looking like in their city. All right. So those are my Jupyter exploits. Let's talk about what I'm currently doing. But before I can really explain it, we have to do a little bit of a neuroscience lesson. So what is a neuron? You can think of a neuron as being a computer, being a very small computer that receives inputs from other neurons, and then just like the software I created, it analyzes those inputs, potentially it updates itself, and then it sends messages to other neurons. Now, 2 neurons, like what you see here, is something that the neuroscience community has really analyzed fully and has a clear understanding of how it works. But when you go up to hundreds of neurons or thousands of neurons, all of a sudden it's much less clear what is going on. And then also you have to add in other cells, because there's other cells other than neurons that also change the behavior of the tissue. Now, until recently, we couldn't grow anything like this that looks all complicated. And also, the only way to then run these experiments was to, cut open the head of an animal and get the neural tissue out of it. But recently, there's been a major breakthrough in, let's say, the last 5 years of the field, where we've created things called cerebral organoids. Now, you can think of a cerebral organoid as being a miniature brain organ from human tissue. And a really cool thing about it is that it really resembles more fully the complexity of different cell types in the brain. And it also resembles the behavior of communication between different cells. Another fantastic thing about it is you can mass produce these things. You can grow tens of thousands of them all at once and really have a factory of neuroscience. So we explained a model that we have, but what exactly is a brain-computer interface? Well, neurons, for the most part, communicate by sending electrical signals between them. And if there's an electrical signal, then if you stick a metal wire into the tissue, you can read the change in voltage. And if you send a little shock of electricity through that metal wire, the neuron will think that you're communicating with it, and you can cause it to send signals to other neurons. So here you see 4 electrodes, electrical wires, receiving signals, and that is what would be a pretty standard neuroscience experiment. However, again, in the last, now let's say 3 years perhaps, there's been a major breakthrough in the field that really changes our ability to see what's going on in neural tissue. And this is caused by high-density CMOS arrays. So in this thing, this is, let's say this is the size of a large cell phone, And then that little chip there, if you zoom in on that, you see this tiny little kind of brownish gray thing. That has about 20,000 electrodes on it. And with that number of electrodes, you can make recordings from, hundreds of neurons. And all of a sudden, you have a much fuller picture of the dynamics of communication between these neurons. For the neuroscientists in the room, our lab builds a lot of its own hardware. This is an optogenetics cable. What that means is that you edit the genome of the neurons so that when you shine light on them, it causes them to change in a certain way. And that is useful for our experiments. All right, let's talk now about how we go from tissue to a computer interface. So we start with the brain tissue, and then we connect it to all the biological devices that people just generally use to keep tissue alive. This is what the setup looks like for one cerebral organoid. Now, again, our lab creates a lot of our own hardware from scratch. So this is a picture of what it looks like when you run this type of experiment on hundreds of cerebral organoids. Now, we then use the Internet of Things to connect all of these devices that we use to a Jupyter website. And this is a website that our lab really just uses on a daily basis. People have the accounts. We use this all the time. Don't be fooled that it looks a little different. This really is just Jupyter. You can see right up there in the upper left-hand corner. And then through that website, there are interfaces for controlling the tissue and defining the experiment. So here on the left, you see an example of what it looks like to control a device, where you select a device, you send a command to it. But then the cool thing is you can also kind of schedule commands. So you can say, hey, we run maybe a week, 2 weeks experiments. We do things that long where not a single biologist is touching anything. You just have the experiment run for 2 weeks, and you are collecting data that entire time. And all of it is programmed. And again, just like everything that I'm showing, this is not like, oh, this is something we are thinking about building. No, this is the actual code. These are websites we go to all the time for running these types of experiments. This is just very common for our lab. Oh, let's see. I want to make sure oh yes, I can safely close that. Okay. Now also for kind of some of the more complicated stuff, like how I was talking about those closed loop machine learning style algorithms, for that you really want to do more nuanced code. So yes, we have these GUIs and widgets for some types of high level experiments, but for those really heavy computational experiments, we built some very good packages that allow us to control these closed loop experiments in a very easily defined way. The data from these experiments also is coming in live. So, as you're running it, you get real-time images automatically. You get real-time images of the different neurons and their communications with other neurons. I'm glossing over this, but that is a massive achievement. To give you an idea, that brain-computer interface for a single cerebral organoid running for 1 day is about 1 terabyte of data. So you run it for 2 weeks, they're talking 14 terabytes of data per organoid that has to be analyzed automatically and given to the user every hour, or let's say every 2 hours. And also we built in fail-safes. So if this experiment breaks, we have code that looks for it, sends emails to everybody. A biologist will run in and try to save things last second. And that has been very useful. The biologist will understand why that is useful. Okay, this is all very impressive. A great question is, why are we even using Jupyter to do this? Why take the extra effort to even do this in Jupyter? And there is a good answer to that question. So first off, Jupyter is a website that anybody can use. But another great thing about Jupyter is that it's very lightweight. So you put it inside of a Docker container, and that same Jupyter code, Docker image that you created, can serve multiple different purposes. So let's start by considering collaborations. Oh, yeah, because purposes that I'll highlight here is sharing data, collaborations, and education. We're going to start by looking at collaborations. So as I said, Braingeneers is not one lab. It's a consortia of many scientists from different universities. There's UC Santa Cruz, we're creating the software, but we're working with Washington University St. Louis. We're working with University of California, Santa Barbara, the Allen Institute, and MIT. And so, again, because this is a website, this stuff can be, like, the experiments we run, someone from MIT can log in, use what we have to design their experiment. Also, they set up Jupyter websites on their place, and then we log in and control experiments remotely. So it's incredible for collaborations. The website right here, so this is our WetAI running at UC Santa Cruz. It's actually running on a server in San Diego called the National Research Platform. That is a supercomputing cluster, so that some of our experiments we need to do 500 GPU neural networks, and we have those resources instantly available to us. Just to highlight some of the experiments happening from other labs, this is some really cool stuff from MIT where we are creating cutting-edge electrode arrays for collecting data, some incredibly cool mouse experiments on sleep cycles, but there is one experiment, one collaboration that I really want to highlight. This is the collaboration that I'm leading at UCSF, and for that, neurosurgeons are extracting human brain tissue from patients on the operating table. And then that tissue is being placed on the brain-computer interface that I showed earlier at the very beginning of this talk. And then we run closed-loop experiments on real human brain tissue. That's never been done. So that is a rather big accomplishment. Now, These experiments, again, they're online. You know, when I arrived in Paris, I found out the day before that there was someone on the operating table. So it's time for me to get to work. So every single day that I've been in Paris, between the hours of 11 p.m. and 3 a.m., I've been running these experiments. So, sorry if I am a little sleep deprived, but it's incredibly exciting. I'm sorry I can't talk any more about the stuff at UCSF. It is confidential. The professors there told me to really not mention anything more than what I've already said about the experiment. Now, I think there's some great opportunities as well, from the stuff that I've just heard in this conference. For example, this is extremely restricted data, extremely restricted to run these type of experiments. Something like Jupyter Lite, because it all runs locally, we can basically have a website where we have all the code set up for running these types of experiments, for doing all the very, very nuanced analysis. And people can go and instantly do the analysis that we can do without ever having to worry about sending the data, because this is type of stuff where you can never upload this data to another server. It is just incredibly secure. So let me now jump into some other aspects of how we use Jupyter. So what happens, after we publish the fancy paper that we are currently writing, and we then want to make our data public? Again, this is very secure data. Maybe we can make some of the ePhys data public, but certainly not the genetic data that goes with it or the patient's record. So for sharing data, we use Terra. And Terra is it's a software created by the Broad Institute, and they've invested heavily in it. You can basically think of it as a Jupyter interface with some wrapper around it that basically allows you to share your code and your Jupyter analysis with others. And what makes it incredibly valuable is it's not just sharing anything. You can share extremely restricted medical data on Terra. And then you select who you want to share it with. All right, so we use the Terra platform as our basis for sharing the data. But again, because this is a lightweight Docker image that has all of our code in it, we can use that Docker image, put it up onto Terra, and now the people who want to analyze our recently published neural data can use all of the very complex analysis and resources that our own lab uses. And the last topic I would like to hit is education. So one cool thing we did is we used this WetAI platform to allow students at a high school that had less resources, giving them a chance to run biological experiments, and that was very fun. But there's something going on, again, at this exact moment that I'm very excited about, because I am, being the TA for this class and also doing many of the lectures for this class, a class called Mathematics of the Mind. Now, in this class, the students actually get to run experiments on cutting-edge tissue models. This is called a connectoid. Again, this is something that's so recent within the field of neuroscience that I think there's one paper about connectoids, but this is so recent there's barely even any research published about these things. So here you see two cerebral organoids, and if you place them close to each other, they'll kind of grow out and create a bridge that connects them. And when that connects, you just see this huge amount of complexity and beautiful neural patterns appear. So we have the ability to mass produce these things, so why not give these to students and have them do their own experiments on this type of tissue? So I released this homework, I think I released this Wednesday or Thursday, before I came here to Paris. I've been teaching them how to do neural analysis, and then this last homework was an introduction to organoid experiments. And in this one, they create their own stimulation pattern, and they decide what messages they're going to send to these cerebral organoids. And then, they have to justify why they want to do the experiment that they want to do. But then, right when I get back to Santa Cruz, that week, I'm going to then run these experiments, and because of how the interface is set up, they will see their experiment run live on the tissue. And so here is a summary of the 3 use cases I've shown for our Jupyter platform as a brain-computer interface, for collaborations, for sharing, and for education. I just want to quickly announce that we are looking for a Jupyter programmer. If there's one thing I learned from this conference is that how I use Jupyter is horrifically out of date, and it's time to get a facelift. So if you are interested in a master's or a PhD in computer science, please talk to me. Or if you recently graduated and you just want to do something super cool for a year and you want to work remotely, we can also do something like that. I will say though, living in Santa Cruz is amazing. It's the closest thing I've ever seen to paradise. Here you see an amusement park right on the beach. This photo right here is not a national park. This is actually just the campus. This is built in the middle of a redwood forest, which is amazing. And I am an avid surfer, so I'm always happy to announce that Santa Cruz is a world-famous surf location. So with that, I'd like to thank everybody who has made this research possible. Our lab is massive, so I can't name everyone. It's over 60 people in our lab. But some of the rock stars who I just work with every single day are Kate, Suri, Ash, and Spencer. My mentors are David Hausler, Sophie Salama, who does a lot of the wet lab work, and Mircea Teodorescu, who does the electrical engineering. And our sponsors, well, at least a short list of our sponsors is the NIH, Schmidt Futures, the National Science Foundation, and the Howard Hughes Medical Institute. Finally, I'd like to thank Jupyter and NumFocus for hosting this conference and for providing the scholarship that allowed me to come here. So thank you very much.
Thank you, Matt. Well, great talk, thanks. Just as a way to start the question, any thoughts about how to the reproducibility of your Jupyter notebooks? Are you going to disseminate organoids everywhere, so to everyone who wants to reproduce?
Yeah. So in terms of reproducibility, the nice thing is everything we do is open source, from the electrical engineering to the hardware. So in theory, people can just reproduce it. Granted, that's a huge amount of work. But in terms of reproducing at our lab, we are growing hundreds of these things. Most people are happy if they do an ePhys, electrical physiology experiment, where it just happens once. We grow hundreds of these things. You run the same experiment hundreds of times on the tissue. So that tackles reproducibility. Now, in terms of sharing it, I mean, if a lab has, the $20 million to kind of get all the biology, first you got to hire people who can grow the tissue, then, we will be happy to provide the hardware. And we do. Like for UCSF, we actually donated, a $70,000 device so that we could get that data set that we wanted from human tissue. So we do collaborate and work with others, either by allowing other people to run their experiments on our platform, or by allowing us and setting up our software at another university to allow us to come in and test algorithms over there.
Hi, I had a question.
Yeah.
First of all, really, really cool talk. It had a lot of new things that I didn't know about. I was wondering if your, new invention in its own way could be used to figure out pathology, or maybe potentially be used in vivo one day?
Yes. So I can't talk about it, but yes. Absolutely, it is actively being used in those ways. That's all I can say.
Okay, I get it.
Sorry.
Thank you for a great talk, really interesting. And I love that you're teaching in high school. This is a really exciting opportunity. Would it be possible, or have you considered doing this virtually so more people have access to live experiments?
Yes, a great question. So Mo, the researcher here. Oh, sorry, it's not there anymore. It's okay, it's okay. But what we have is we are actively now, I think we worked once with a local high school. Now we're expanding it. I think it's 4 colleges in Latin America from Bolivia to Mexico. And then we're allowing them to, again, run these types of experiments. So that, I think, will be happening in September. Is when that will start.
Anyone else? Any questions? Ah, yeah. One. Yeah.
Do these organoids think?
I mean, no. Short answer, no. So they show neural behavior, and they're miles ahead of what existed previously in biology. Before that, it was kind of a 2D cell culture, and it's basically just growing neurons and hoping they connect in a way that was reasonable. These are much more complicated, and they resemble the dynamics of the brain much better. However, they only exist for maybe, I think, 120, 150 days. They are extremely limited in terms of any notion of consciousness. It's so far away from that I don't think we need to worry about it. At least in the near term. That is not something we are concerned about. There's a reason why the serious stuff happens on real brain tissue. The organoids are a great model of behavior for starting out, but they're not near true brain tissue yet.
Yeah. Okay. Thank you very much, Matt. That's a very nice way to end up the talk and the session. So I would like to thank again you and all the other speakers for this very great session. If you can, kind of close.